Most of the world didn't have it until colonization started in the 15th century.
And it was discovered that the rhesus factor could be positive or negative. Most humans in the world are rhesus positive.
For years, type O blood has been treated as a simple medical fact, just another detail in the charts, a box to tick during a donation drive. But behind this unassuming label lies a mystery that has quietly defied explanation. Around the world, patterns in its presence tell a story that doesn't quite add up. Some populations cling to it with an intensity that science struggles to explain. And now, new research is forcing experts to reconsider everything they thought they knew about this rare genetic signature. The blood that wouldn't go away. In the world of medical science, blood typing is often presented as a simple chart, A, B, AB, or O, with a few notes about compatibility for transfusions. Among these, type O stands out for one remarkable reason. It carries no A or B antigens on the surface of its red blood cells. This absence is what gives it the title of universal donor. When type O blood is transfused into someone with a different blood type, the immune system is far less likely to see it as an intruder. It slips quietly into the recipient's system, delivering oxygen without triggering a dangerous reaction. Doctors have relied on this biological blank slate for decades in emergencies, where time is short and matching blood types is impossible. In such moments, type O has saved countless lives. Yet, beyond the emergency room, this apparent neutrality hides something unusual. As scientists expanded their research beyond hospitals and into the realm of population genetics, they began noticing a pattern that was as fascinating as it was puzzling. Across vast regions of the Americas, from the frozen tundra of Alaska to the humid depths of the Amazon rainforest, type O wasn't just present, it was dominant. In some indigenous communities, it wasn't merely the majority blood type, it was nearly the only blood type. Studies of certain Amazonian tribes revealed that close to 100% of their members carried type O. In remote Andean villages and Arctic hamlets, the story was much the same. Generations upon generations showed almost complete uniformity in this one trait. At first glance, the scientific community had a convenient explanation for this. Isolation, they argued, can lead to something called genetic drift, the random fluctuation of genetic traits over time. In small, isolated populations, certain alleles, those traits will remain prevalent in their descendants. These ideas seem to fit neatly with the story of type O dominance in remote communities. After all, if the original settlers of these regions happened to carry type O, and if they remained cut off from outside populations, it wasn't hard to imagine how the trait could persist for centuries. But as more genetic data poured in from across the Americas, the by-chance theory began to falter. The pattern was simply too consistent. It wasn't just one or two isolated groups that showed high frequencies of type O. It was nearly all of them. And the uniformity went beyond what could be expected from random drift alone. Even in populations that had experienced some contact with outsiders, the dominance of type O held firm. In certain cases, other blood types weren't just rare, they were entirely absent. This level of purity raised difficult questions. Let us consider that genetic drift was the only factor, and then we could at least expect to find some pockets of variation here and there, traces of other blood types remaining in the mix. Instead, the data indicated that an intentional filter has been used, as if something very far back preserved type O, while A and B were erased entirely from the genetic record of whole regions. For years, the implication was quietly swept aside. It was more comfortable adhering to old explanations rather than suggesting that there might be a deeper and more disquieting reason for the pattern. But over time, this avoidance became impossible. As archaeologists began to unearth remains from far back in ancient history, and geneticists built tools that could yield DNA sequences from bones thousands of years old, the tale of Type O's ultra-domination only deepened the riddle. The phenomenal scale and uniformity of the phenomenon could be taken to mean that it was not an accident of history. Rather, it was a signal, hinting at much older, much stranger origins. That is when researchers began to search back further, looking not just into present-day populations, but into the migrations of humanity itself. The more they traced the path of Type O, the more the familiar story of how people first came to the Americas began to fracture under the weight of new evidence. But the more scientists looked at where Type O came from, the more the ancient migration story began to crack. Cracks in the ice bridge theory. For decades, anthropology textbooks told a single, tidy story about how the first humans arrived in the Americas. Roughly 15,000 to 20,000 years ago, during the last ice age, sea levels were far lower than today. The Bering Strait, now a narrow stretch of frigid water between Siberia and Alaska, was once a wide land bridge known as Beringia. According to the traditional theory, small bands of hunter-gatherers crossed this frozen expanse, following herds of game into a new and uninhabited world. Cut off from their Asian homeland by rising seas and melting ice, these pioneers became the ancestors of all indigenous peoples of North and South America. The genetic picture seemed simple enough. If this migration involved only a small number of people, perhaps just a few hundred, they would have carried only a portion of the genetic diversity present in their original Siberian population. Over thousands of years, isolation would amplify those original traits, explaining why certain genetic markers became dominant among indigenous peoples. By this logic, the overwhelming prevalence of Type O blood in the Americas could be explained if the founding migrants just happened to carry mostly or entirely Type O alleles. It was a neat, self-contained theory that lined up with the tools and evidence available at the time. But then came the revolution in ancient DNA research. As scientists learned how to extract and analyze genetic material from skeletal remains thousands of years old, they began looking closely at the blood type alleles carried by ancient Siberians, the very people who, according to the theory, had crossed the land bridge into the Americas. The results didn't fit the old narrative. Far from being genetically uniform, these ancient Siberians displayed a wide variety of blood types. Type A, Type B, Type O, all were present in significant numbers. This was not a small, genetically limited population dominated by Type O. It was a rich, diverse mix, much like what we see in Europe, Asia, and Africa today. The implication was clear. If the first Americans came from this population, they would have carried the same variety of blood types across the land bridge. This is where the problem began to emerge. If multiple blood types had crossed into the Americas, why are they not still present in indigenous populations today? Where did the A and B alleles go? Their complete or near-complete disappearance is not what we would expect from random genetic drift. Drift can reduce diversity over time, but it rarely wipes out major alleles entirely, especially not across entire continents. Rather, the genetic evidence appeared to point toward something much more dramatic, often referred to as a bottleneck, a collapse in blood type diversity after the arrival of humans in the Americas. The A and B alleles did not merely diminish, they became extinct in many areas, leaving Type O as their only survivor. This was not a monotonously slow, random walk of genetic drift. Rather, it was sudden, decisive, and essentially synchronous. Such a pattern raised troubling possibilities. Was AB blood group type simply eliminated by some calamity that struck the early populations? Was it a pathogen that selectively targeted those blood types? conferring a selective advantage to type O? Or was some other, as yet unknown selective force acting on the population, changing the genetic landscape in just a few generations? For whatever reasons, this nice and clean story of migration could no longer hold. The vanishing of A and B in the Americas was not the sort of genetic change that could be readily explained away by reference to geography, climate, or chance. It was the signature of an event, a powerful filter through which only one blood type passed intact. The evidence was beginning to suggest that the journey into the Americas was not just a migration, but the beginning of a survival trial, one whose scars are still visible in our DNA today. And what scientists found in ancient remains suggested this disappearance wasn't slow. It was deliberate. The vanishing bloodlines. By 2025, the mystery of type O's dominance took a sharp and unsettling turn. A coalition of geneticists from Canada, Brazil, and South Korea launched an ambitious project to map the history of blood type alleles in the Americas using ancient DNA. Their approach was unprecedented in scope, sequencing genetic material from skeletal remains spanning thousands of years, recovered from Alaska to Patagonia. The goal was simple: trace the presence of type A, B, and O alleles across time and geography to understand how today's patterns came to be. The results were anything but simple. Early remains found in Alaska, close in both time and location to the first crossing from Siberia, told an unexpected story. These ancient Alaskans carried not just type O, but faint traces of type A as well. This alone was a breakthrough. It proved that the first peoples arriving in the Americas did indeed bring more than one blood type with them. The old theory that only type O had crossed the land bridge was no longer viable. But as the researchers examined remains from sites further south, the traces of type A began to vanish. In skeletal remains from the central plains of North America, type A was absent. In samples from Mesoamerica, gone. By the time they reached the remains of early Andean peoples in South America, there was no sign of A or B at all. Across thousands of miles, the story was the same. Type O alone endured. This abrupt disappearance defied the slow, uneven patterns of genetic drift. It looked less like a gradual fading and more like a purge. Something, or someone, had removed A and B from the gene pool with ruthless efficiency. The researchers began to speculate. Could a catastrophic epidemic have swept through the Americas, exploiting vulnerabilities in the A and B blood types? Certain pathogens are known to bind to blood group antigens, using them as entry points into the body's cells. If such a pathogen targeted A or B antigens, type O carriers would have enjoyed a survival advantage. Other theories were equally grim. Perhaps a previously unknown virus or bacterial strain, now long extinct, once burned through ancient populations, sparing only those with the genetic makeup to resist it. Others suggested environmental factors could have played a role, with extreme climate shifts or nutritional stresses interacting with immune systems in ways we still don't understand. Yet all of these scenarios pointed toward the same conclusion: something highly selective had been at work. The most telling clue came not from the body of the study, but from a single easily overlooked sentence in its abstract. The data imply a directional selection event, potentially immunological in origin, favoring O alleles over all others. In plain language, this meant that the changes in blood type distribution weren't random. They followed a direction, a strong push by evolutionary forces toward type O. And those forces were almost certainly linked to immunity and survival. This was a sobering realization. It suggested that the near-total dominance of type O in so many indigenous populations wasn't just the product of geography or chance. It was the fingerprint of a survival trial that had swept through the Americas in deep prehistory. a trial so severe that it left only one blood type standing in vast swaths of the continent. If this was true, then type O was more than a medical convenience. It was the genetic legacy of those who had lived when others perished, the blueprint of survival itself. If type O survived when others didn't, what hidden advantage was it carrying in its very code? The immunity rift. When scientists began to explore what made type O blood so dominant in certain populations, the answers started pointing toward something more than genetic happenstance. Patterns emerged in global health data, patterns that hinted at a quiet resilience hidden in plain sight. People with type O blood across different continents and environments consistently showed lower rates of severe illness from some of history's most dangerous diseases. Cholera, malaria, dengue fever, even certain strains of severe respiratory infections all seem to exact a slightly smaller toll on type O carriers. This wasn't to say type O individuals were immune. They could still contract these illnesses, sometimes even fatally. But when survival odds were measured over large populations, type O appeared to have an edge. It was a subtle advantage, not enough to protect everyone, but enough that, over generations, more type O carriers survived outbreaks. The more researchers compared modern survival rates to historical disease patterns, the more it became clear that this quiet edge might have been critical in the distant past, when medical intervention didn't exist. The most striking evidence came from the highlands of the South American Andes. Here, in some of the harshest living conditions on Earth, communities had not only endured for centuries, but thrived. The air is thin, ultraviolet radiation from the sun is intense, and exposure to infectious disease has been constant for generations. Yet, many of these communities enjoy a surprising level of long-term survival. When geneticists examined their blood, they found something remarkable: an unusually high prevalence of a gene variant known as IL-17 variant 52C. This small change in the immune system's blueprint affects how the body responds to infections. In many severe illnesses, death doesn't come from the pathogen itself, but from the immune system's overreaction, a cytokine storm that causes widespread tissue damage. The IL-1752C variant seems to act like a biological thermostat, keeping immune responses strong enough to fight infection, but controlled enough to avoid self-destruction. What made this discovery even more significant was its strong association with type O blood. In these Andean populations, type O and the IL-1752C variant appeared together far more often than chance would predict. For eons, this dreary stuff hinted that something had been checking the populations continuously and eliminating those which had failed to strike the right balance with their immune systems. Surviving with their type O blood and the IL-1752C variant, they were more likely to survive, thus passing their genes into the next generation. By pure chance alone, the odds against this are small. It involved many eons-long tedious history of selective pressure and many repeated survival struggles, possible against pathogens whose presence today is unknown. Whatever it was, it pushed human biology into a corner, promoting individuals with type O blood across this immune variant to remain quietly resilient in its grip. These results were not published in lots of places. Such studies remained confined within specialist journals that were mainly readable by the experts. There was none of such news on the evening headlines, as has previously been said, that a certain blood type might carry echoes of ancient survival trials. Evidence thus piled up for those paying attention. Type O wasn't just a universal donor. It might be the living memory of a time when only the most adaptable immune systems survived. And as scientists began to search for more clues, one discovery stood out above the rest. It came from the frozen ground of Siberia, preserved for over 12,000 years, and it carried within it a mutation unlike anything researchers had seen before. And then, frozen deep in Siberia, an ancient skull revealed a mutation unlike anything seen before. The Lake Bol marker. In the remote frozen stretches near Lake Bol in Siberia, archaeologists uncovered a find that would deepen the mystery surrounding type O blood. Beneath layers of ice and soil, preserved in the permafrost for more than 12,000 years, lay the skull of an ancient human. The conditions had kept the remains in remarkable shape, and geneticists knew this was a rare opportunity, the kind of discovery that could offer a direct glimpse into the bloodlines of humanity's distant past. When the DNA was sequenced, researchers found something extraordinary. The individual carried a rare genetic marker in the ABO blood group gene cluster. This was not a typical mutation, but a deletion, a section of genetic code missing entirely. Scientists named it O-delta variant 1. Deletion mutations of this kind are far less common than single-letter changes in DNA, and they usually signify that some extreme evolutionary pressure has forced the body to adapt in a drastic way. What made this finding even more fascinating was its exclusivity. O-delta variant 1 was not present in European DNA. It was not in African or East Asian populations. Even within Siberia, it was absent from most ancient samples. The only place this genetic signature appeared was among certain indigenous groups in North America, a clear link between the ancient Siberian individual and the descendants who would later populate the Americas. For geneticists, this was like finding a unique fingerprint stamped across millennia. The deletion appeared to have a functional purpose, one that may have influenced how the immune system recognized threats. It suggested that, at some point in prehistory, this lineage faced an intense, possibly life-or-death challenge that reshaped its genetic code. The implication was staggering. This wasn't just random variation. This was the remnant of a biological battle. Something, whether an environmental toxin, a long-forgotten disease, or an immune-triggering catastrophe, had left such a deep mark on this population's biology that it altered their genetic structure in a way still detectable today. And it wasn't a general human adaptation. It was specific, belonging to a single bloodline that had survived while others disappeared. Even more intriguing was how tightly O-delta variant 1 was associated with type O blood. In modern indigenous groups where this variant appeared, type O was overwhelmingly dominant, sometimes to the point of exclusivity. This wasn't just about who crossed the Bering Land Bridge. It was about who survived long enough to pass their genes on. For anthropologists, this discovery added a layer of urgency to the mystery. If the same genetic marker could be traced from an individual living in Ice Age Siberia to living communities thousands of miles away, it meant we were looking at an unbroken chain of survival. Across climate shifts, migrations, and catastrophic events, this trait had endured. Yet, as significant as this was, it was still only a piece of the puzzle. It told us there had been intense pressure on early human populations, and that this pressure had produced lasting genetic changes. But it didn't tell us exactly what the threat had been, or why type O blood, in particular, seemed to carry so many of these survival signatures. That answer would come from an entirely different discovery, not in the frozen tundra, but in a modern DNA sample submitted in 2025. This case would reveal something no one expected, a possible built-in defense switch buried deep in the human genome. But in 2025, one man's DNA sample uncovered something even stranger, a possible hidden defense switch. The Crawford Revelation. In early 2025, a quiet entry into a genetic database set off a chain of events that no one could have predicted. It began with Thomas Crawford, a resident of the Blackfeet Nation in Montana, who agreed to participate in a large-scale ancestral mapping project. Like many volunteers, he expected the results to simply confirm his heritage and provide a detailed picture of his lineage. But when his DNA sample was analyzed, it revealed something far beyond routine ancestry. Crawford carried the O-delta variant 1, the rare deletion mutation first identified in the 12,000-year-old skull near Lake Bole. That alone made his sample noteworthy, linking him to one of the most ancient and exclusive genetic signatures known in the Americas. But it was what appeared alongside O-delta that caught researchers completely off guard. In the sequence of DNA surrounding the human leukocyte antigen, HLA region, a critical hub for controlling immune responses, they found an unusual fragment of genetic code. For decades, this segment had been dismissed as junk DNA, a term used for sequences with no known function. But when advanced immune modeling simulations were applied, the fragment behaved in a way that defied expectations. Under normal conditions, it lay dormant, silent in the genome. But when the model simulated extreme biological stress, scenarios involving multi-organ infection combined with oxygen deprivation, the sequence switched on. Not partially, not weakly, but like a light being thrown in a darkened room. Once active, it appeared to regulate a cascade of immune defenses, stabilizing inflammation and protecting vital organs from collapse. In essence, it acted as an emergency survival trigger, only activating in the most life-threatening situations. The researchers had never seen anything like it. It was as though an ancient backup system had been written into the genetic code, a hidden defense that might have been the difference between life and death during catastrophic outbreaks. And yet, this mysterious sequence didn't appear in every human genome. It was disproportionately present among type O carriers in certain indigenous populations, often alongside O-delta variant 1. As the team debated the implications, an even stranger twist emerged. According to a whistleblower from within the Human Genome Archive, a massive international repository of genetic data, this very sequence had been identified before. The source claimed it was first flagged in 1998 during the early phases of the Human Genome Project. At the time, its function was unknown, but its unusual structure had drawn attention. Then, inexplicably, the information vanished from public datasets. The whistleblower alleged it was deliberately buried, never discussed in published research, and excluded from major genetic databases. Why? No one outside the inner circle seemed to know. Perhaps the discovery was deemed too speculative at the time. Or perhaps the implications that some human bloodlines carried a unique, potentially engineered survival mechanism were considered too controversial to release. Whatever the reason, it had taken nearly 3 decades for the fragment to re-emerge, this time tied directly to the type O blood mystery. For the scientists working on Crawford's case, the find was both thrilling and frustrating. They had a tangible link between type O blood, ancient survival events, and a hidden genetic defense system, but no clear understanding of how it had come to be, or why it appeared only in certain populations. One thing, however, was certain. The trail of type O's story was no longer confined to the Americas. New genetic comparisons hinted at connections far across the Pacific Ocean, linking survival codes between peoples separated by thousands of miles of open water. And when scientists traced type O beyond the Americas, the trail led across the Pacific to a stunning connection. The Polynesian Passage. The final piece of the puzzle came from one of the most remote inhabited places on Earth, Rapa Nui, better known as Easter Island. Located thousands of miles from the nearest continent, its isolation has long fascinated historians and archaeologists. In 2025, Excavations on the island unearthed skeletal remains dating back roughly 900 years. These bones, preserved in the island's volcanic soil, were sent to genetic laboratories for detailed analysis. The results startled researchers. Not only did the remains show an overwhelming prevalence of type O blood, but they also carried the IL-17 variant 52C, the same immune-modulating gene variant found in highland Andean populations thousands of miles away in South America. This was no trivial match. The odds of the same rare immune variant appearing in both locations purely by coincidence were astronomically low. The most likely explanation was direct genetic exchange, which in turn suggested contact between Polynesian voyagers and South American peoples centuries before Europeans arrived. For years, the idea of pre-Columbian contact across the Pacific had been debated in academic circles. The evidence had mostly consisted of similarities in crops, artifacts, and oral histories. Now, the DNA itself was speaking. It wasn't just cultural exchange, it was biological exchange, encoded in the blood. If Polynesian navigators had indeed reached South America, they might have carried not just goods and ideas, but the very genetic tools for survival against deadly diseases. Meanwhile, another line of research was taking shape high in the Andes. Archaeologists studying the teeth of ancient Andean individuals found tiny traces of antibodies locked in dental enamel, microscopic records of immune battles fought during their lifetimes. Some of these antibodies were tuned to pathogens modern science has never encountered. They likely belong to diseases that are now extinct, long erased from human experience. What made the discovery even more haunting was who carried these immune markers. Nearly every case came from individuals with type O blood. The other blood types were nearly absent from the archaeological record in these contexts, not because they were overlooked, but because those who carried them may not have survived the disease waves that swept through ancient populations. This painted a grim but revealing picture. In times of severe epidemic or environmental stress, type O wasn't just the most common blood type, it may have been the only one able to endure. Whether through natural immunity, genetic resilience, or sheer adaptability, it repeatedly outlasted its counterparts. And each time, it became more entrenched in the surviving gene pool, tightening its grip on the future of those populations. The Polynesian connection added a new dimension to this survival story. It suggested that the spread of type O and its associated immune advantages wasn't confined to land migrations across ice bridges. It could also travel vast oceans, carried intentionally by human hands and hearts. Those voyages may have been acts of exploration, trade, or alliance, but in hindsight, they also served as lifelines, transmitting the genetic tools that had once saved entire populations from extinction. In the end, what began as a simple question about blood type had expanded into a sweeping narrative of contact, catastrophe, and endurance. The genetic footprints of type O blood can be found from the jungles of the Amazon to the volcanic soils of Easter Island, from Andean highlands to Siberian permafrost. Each discovery added weight to the idea that this blood type is far more than a clinical classification. It is the product of countless survival trials, encoded in the veins of those who carry it. And together, these clues paint a hidden history of collapse, survival, and an unbreakable code flowing in our veins. Type O blood may be far more than a universal donor. It could be humanity's ultimate survival signature. If the science is right, it carries the memory of a near extinction, a genetic whisper from a time when only the strongest endured. The question now is, if your blood holds such a secret, what will you do with it? Like, share, and subscribe for more deep dives into the science and history they don't teach you. And check out the next video on screen for more hidden truths about our past.